Michelle completed her bachelor's in biological sciences at the University of Bath and subsequently completed her PhD at NIAB and the University of Reading in the labs of Richard Harrison and Robert Jackson. Her PhD focused on host specificity of Pseudomonas on fruit trees using comparative genomics. And Michelle continued that work at NIAB and focused on phage-mediated horizontal gene transfer and evolution. And then I got to know Michelle actually when she was here at the Sainsbury Laboratory in the lab of Wenbo Ma, where she was focusing on bacterial plant pathogen effectors and how they manipulate metabolism. So now Michelle is an assistant professor at Michigan State University, where she is leading a group and continues looking at the genomics of plant pathogenic bacteria. So with that, I'll hand over to Michelle. Thank you very much.
All right, thank you so much, James. I'm just going to preempt that we have a thunderstorm going on here in Michigan, so if you hear any thunder, I apologize. But yeah, thank you so much for the invitation to present. I really think that the GetGenome initiative is really exciting and a really good initiative to support global research in bacterial genomics. So today I'm going to talk to you about some research which is going on in my new lab, which I started last year, looking at bacteriophages and how they may be involved in enhancing bacterial virulence on plants. Okay, so in my lab, we work on bacterial plant pathogens, and here we mainly work on horticultural crops, and this is a picture that I took last year in a cherry orchard in Michigan, and you can see that generally these plants are quite healthy. However, if we look at the plant at the front of this image, we can see that this whole shoot of this cherry tree has completely died back, and this is due to a bacterial plant pathogen called Pseudomonas syringae. And the Pseudomonas lives epiphytically on the surface of the plant during the year, and under certain conditions, it will enter the plant and cause disease. And I'm really interested in my group in understanding these epiphytic populations and using genomics to understand what drives the emergences of disease-causing organisms. So if we zoom in to the surface of a leaf, what we find is that these pathogens and non-pathogens exist in diverse populations on the surface of leaves, and these are in high population densities, often exceeding over 10 million bacteria. And I'm interested in understanding the factors that trigger the emergence of pathogens and the factors that alter the evolution of these different bacterial communities. So, for example, here are just some different factors. So these different communities are exposed to different abiotic factors. They're exposed to UV light, which is a major stress on the bacteria, different nutrient conditions. They're also having interactions with each other. So the bacteria maybe are antagonistic or in competition with each other. So there's these microbe-microbe interactions. Also, the plant immune system plays a major selective pressure on these populations. So the plant has its own immune system, which is influencing the evolution of these bacteria by having an immune response, which can kill these bacteria on the surface. But what we're really interested in are bacteriophage viruses. So these are viruses that infect bacteria and they kill these bacteria. So these viruses have major influence on the population dynamics of bacterial populations. So by killing bacteria, they can cause major shifts in which bacteria, how the populations go up and down. And this happens across all environments around the world. They also are able to pick up genes from bacteria when they infect. And so they are key mobile genetic elements, and they are involved in horizontal gene transfer between bacteria. And these bacteriophages, or phage for short, have distinct infection life cycles. So one of these is the lytic life cycle. So when the bacteriophage infect, they will latch onto the bacterial cell and they will inject their genome into the bacterial cell. They will then use the host cell machinery to replicate their genome and produce new phage particles, which when there's enough of them will lyse the bacterial cell, and these phages will move to alternative hosts and infect them. So this is the lytic cycle. An alternative cycle is the lysogenic cycle. So this is where when the phage infects the bacteria and it injects its genome, this genome gets integrated into the bacterial genome and becomes a dormant prophage. And this prophage, what happens is all of the genes that are required for producing new phage particles get switched off. The prophage is then passively replicated with the bacteria during their cell division, and it's maintained in the population in this way. And really interestingly, when these prophages can actually be reactivated to go back into the lytic cycle in a process called induction, and this is often when they perceive that the bacterial cell that they're within is under stress, and they activate to produce new phages, which can then move to alternative hosts. And during this replication process, as I mentioned, the bacterial DNA can actually get integrated into the phage genome. So when they're replicating and producing new DNA molecules, they can actually sometimes pick up bacterial genes. And when they pick up these bacterial genes, they can then act as agents of horizontal gene transfer. So in this way, these phages are able to influence bacterial evolution by moving bacterial genes around the bacterial population. And we call the genes that they take up into their genome accessory genes or moron genes, because these are not genes required for producing new phage. They're genes that are actually of bacterial origin, and they can influence bacterial phenotypes. And it's been found that the genes that the phages contain often have very important consequences for the bacterial in certain conditions, and they can radically change the phenotype of the bacteria. For example, phages can acquire genes that can alter the pathogenicity of the bacteria. For example, they can acquire genes that are secreted proteins or toxins, which make the bacteria more pathogenic on their host. They can also carry genes which are involved in defense systems, and these are really interesting systems which are bacterial immune systems that provide the bacteria with defense against other phages. So this is a really emerging field looking at these defense systems, because a bacteriophage can actually provide defense for its bacterial host against other phages, so they can carry these defense systems. And finally, they could also carry, for example, genes that provide resistance to antibiotics or other antimicrobials. So these phages, by carrying these genes, can provide bacteria key fitness benefits in different or certain conditions, and therefore they're very important in influencing the evolution of these bacteria. And I'm just going to tell you about two really interesting examples as a biologist of how phages can really radically change bacterial evolution. So one of these is E. coli, found in E. coli, and there are phages in E. coli which carry the Shiga toxin. And this Shiga toxin is required for the E. coli to be pathogenic on humans and cause disease in the human gut. And it's thought that the acquisition of the Shiga toxin on this phage in certain E. coli lineages has led to major epidemics in human disease. Another really interesting example is Wolbachia. So Wolbachia is an endosymbiont of insects, and it's a really interesting bacteria that can manipulate the sex determination of the insect. So this bacteria can change whether this insect will become male or female. And what's been found is that the genes that are required for this sex manipulation are often encoded on phages within Wolbachia. So therefore, the phage, by carrying these sex determination genes through the bacteria, is actually influencing the sex of this insect. And we call these extended phenotypes, because basically the phage is indirectly, through its bacterial host, influencing another organism. So it's just a really cool example of biology as a researcher. So I'm really interested in bacteriophage interactions in terms of how they influence whether a bacteria is pathogenic on a plant. So we have these kind of tripartite interactions where we know quite a lot about how bacteria cause disease on plants, but we don't know very much about this relationship between the bacteriophage and the bacteria and how this influences pathogenicity evolution. And we're studying this in Pseudomonas syringae, and Pseudomonas syringae is a species complex of bacterial plant pathogens which infects over 300 different host plant species. And here are just some of the symptoms that it causes. You can see it causes these cankers on trees, but it also infects and causes disease on leaves as well as fruit. And most people know Pseudomonas syringae because of the well-known pathosystem called PST-DC3000, which infects Arabidopsis. And this disease of Arabidopsis caused by Pseudomonas syringae has been fundamentally important in improving our understanding of how first the plant immune system works, but also how bacteria are able to cause disease. And in order to cause disease, Pseudomonas syringae uses its type-free secretion system, which is a large molecular complex which the bacteria use to inject effector proteins into plant cells. So this bacteria has gone up to this plant cell and it injects things called effectors, which are proteins that work within the plant cell to suppress the plant's immune response and promote conditions that favor disease. Therefore, these effectors are very important for enhancing the virulence of Pseudomonas on plants. However, during coevolution, plants have acquired resistance genes or NLRs, which can actually directly or indirectly detect the presence of these effectors and trigger an immune response. And therefore, sometimes, depending on the plant that the Pseudomonas is infecting, these effectors can actually be avirulence effectors because they trigger an immune response and make the pathogen less able to cause disease. And it's thought that the particular complement of effectors that a bacteria has will dictate whether or not it can cause disease on a particular host plant species. So they need to keep the effectors that are important for causing disease, but they also need to lose or mutate those effectors that trigger immunity. And if we look at Pseudomonas syringae, as I said, infects many hundreds of plant species. However, individual strains or clades are often host-specific, and this means that they're only able to cause disease on one or a few hosts. And therefore, what I've always been very interested in is how the bacteria can rapidly change their effector repertoires to cause disease on a new host. So how do they adapt onto a new host, and how do they rapidly change the effectors, keeping those that are important for causing disease while losing those that are triggering immunity? And Pseudomonas syringae has 84 different effector families, and each strain possesses between 10 and 40 of these, and they can rapidly reshuffle them. So one thing that we wanted to understand is if these effector repertoires can be shaped by mobile genetic elements such as bacteriophages, and if this allows the bacteria to adapt onto new hosts. So in this talk, I'm going to go through two main projects that are ongoing in my lab that are kind of interconnected. The first is using bioinformatics and genomics to understand how common prophages are across Pseudomonas syringae and if they carry virulence-associated genes such as type-free effectors. And the second one, I'm going to give you a specific example of how these prophages could contribute towards host adaptation and virulence. So the first question we wanted to ask was, how common are prophages within the genomes of Pseudomonas syringae, and could they therefore be playing an important role in Pseudomonas syringae's evolution as a plant pathogen? So I've put a kind of flow chart on how we do this. We're doing a very large phylogenomic study. So what we did what we first did was we collected all of the Pseudomonas syringae genomes from NCBI, the online database of genomes, and we had about 5,000 Pseudomonas syringae genomes. However, those of you that work with bioinformatics know that the more genomes you have, the harder it is to do computational work, and it just gets more and more intensive. So what we did is we then decided to remove strains that were almost identical to each other, so only picking one representative. And in order to do this, we use a program called FastANI, which looks at the average nucleotide identity of all of the genomes in common with each other. And then I removed strains. For each clonal group, I would only keep one strain. So this allowed us to narrow down this set of genomes to just over 1,600 genomes. We then do gene annotation using a program called BACTA in order to look at what genes the bacteria have. We then do pangenomics, which is where we take all of these genes and see and group them into homologous groups. And we use this program called PANARU. We then build a maximum likelihood phylogenetic tree with a program called IQTree. And this can allow us to do phylogenetic analysis. And here we have a large phylogenetic tree of Pseudomonas syringae, and this contains over 1,600 strains. And what I've highlighted here is all of the different kind of phylogroups, or 95% identity kind of species, as we call them, across the phylogeny. And this covers the diversity of all of the known Pseudomonas syringae that have been genome sequenced. We then use bioinformatics predictions programs such as PhySpy and Fastis to predict prophages within the genomes of each of these strains. So when I did this, I was able to predict over 2,700 prophages across Pseudomonas syringae. And what was quite striking is that this is a graph which shows the number of prophages in each genome on this phylogenetic tree. What was quite striking was that 98% of Pseudomonas syringae possess prophages. So this indicated that prophages are universally present in Pseudomonas syringae genomes. So they're not very rare, they're actually found across the whole of the species complex. And what you find is that some strains, for example, those here, actually have up to 6 prophages in their genome. And because bacterial genomes are quite small, possessing 6 prophages means that actually quite a large proportion of this bacteria's genome is of prophage origin. The next question was, with all of these prophage sequences that we predicted, do they carry virulence genes such as type 3 effectors? So in order to do this, I use tBLAST-N, where I take known effectors from the effector families in Pseudomonas syringae, and then using this tBLAST-N program, I looked within the prophage regions that we had predicted and to see if any of these effector families are present. And what this shows is that we had 23 different effector families, which is each of these kind of vertical columns, were present within prophages in Pseudomonas syringae. And you can see that, for example, this group of strains here, they actually possess 4 or 5 prophage-encoded type 3 effectors. So this indicated that, yes, prophages are present across Pseudomonas syringae, and they can carry multiple different type 3 effectors. So they could be involved in shuffling type 3 effectors around during host adaptation onto new plant species. Our next question was, so I wanted to look further into the genetic diversity of these prophages. Are all these prophages very similar to each other, or are they divergent? And also, could they mediate horizontal gene transfer? Do they have all of the predicted genes to produce phage particles, and could they be able to move around in the species complex? So in order to do this, I took all of these 2,700 prophage sequences that we had predicted from the Pseudomonas syringae genome, and then I acquired over 4,000 Pseudomonas phages from NCBI. So these are bacteriophage genomes that are available online that have been either isolated from Pseudomonas or if they're potentially in the Pseudomonas genome. And then I use a program called VipTree, which takes the proteome of all of these, the predicted sequences, and then builds a kind of large phylogenetic tree based on the similarity of their proteomes. And what we found is that the Pseudomonas syringae prophages present here in the blue are interspersed with other Pseudomonas phages, and you can see them around the phylogenetic tree. And this indicated that these prophages are relatively diverse. They're not all the same prophages. And we found that they all belonged to the class of viruses, double-stranded DNA viruses called the Cordoviricites. And then using hierarchical clustering, we could, in R, we could cluster these into different genetic groups based on being in similar parts of the tree. And you can see that at least 4 or 5 different kind of genetic groups of phage are found in Pseudomonas syringae. The next question was, which ones of these carry type 3 effectors? So what I've highlighted around the edge of the circle here is those phages on this phage phylogeny that carry type 3 effectors. And what you can see is that 4 of these genetic groups can carry type 3 effector genes. And here are some of the type 3 effector genes that we found. So you can see that we have HOP-AR present, and this is an effector I'm going to talk about later in my presentation. This HOP-AR effector gene is actually present in multiple different phages across the phylogeny, and this was quite intriguing, indicating that the same effector family is being picked up multiple times by different phages within Pseudomonas. This group of prophages over here seemed quite different, and they didn't carry any type 3 effector genes. So they seem to be working maybe in quite a different way to influence the bacteria's evolution. The final question we had was, do these do these prophages contain all of the genes required to produce functional phage particles? So in order to do this, we use a program called Phages to predict the phages within the genomes. And this program is a really nice program. It's actually available online if you wanted to use it. And what it does is it takes all of the genes in the phage and then sees if that phage, predicted phage, has the genes required to produce kind of all the parts of the phage particle. So does it contain the tail, the capsid, the head, for example? And then it then gives you a prediction as to whether or not this phage could be intact. And what I've done is I've highlighted those that are predicted to be intact around the edge of the phylogeny with these black squares. And you can see that the vast majority of Pseudomonas syringae prophages were predicted to be intact. And why is this important? This is important because they therefore could have the ability, they're not degraded phage, they're not just a couple of phage genes in the genome, they actually have the fully functional group of genes, which maybe means that they could move. And obviously this is all bioinformatics prediction and would need to be validated with further work, but it's very interesting that most of them seem to be predicted to be intact. Our final question was, so moving away from just the type 3 effectors, we also wanted to look within these prophages in Pseudomonas syringae to see what other genes they maybe contain, what other bacterial genes or accessory genes that could have functional importance for the Pseudomonas syringae species complex. So in order to do this, I took these 2,700 prophages, and then I annotated the genes on these prophages. And then with these annotations, what we did is we can remove those genes that were labeled as structural proteins, so genes that are of phage origin. And then we can then look at what functional groups we have left over, which are our potential bacterial genes or accessory genes. And we can create a word cloud to see what potential other functions these prophages have in Pseudomonas syringae. And here we have a word cloud out of all of these annotations, and with the larger words indicating the like kind of largest functional group. And it was quite annoying but exciting that the largest group was hypothetical. So this indicates that this protein doesn't have a known function, and therefore this means that these prophages in Pseudomonas syringae have a lot of unclassified genes, which could have be potentially interesting for future work. The second largest group was actually transcriptional regulation, and I'm going to go back to that at the end of my talk, because it's quite interesting that these prophages seem to carry a lot of bacterial transcription factors and therefore could be regulating the transcription of other genes in the Pseudomonas syringae genome. And in terms of virulence, we didn't just find type 3 secretion system genes, but we also found genes that maybe influence bacterial adhesion to host cells, bacterial motility, so the ability of the bacteria to swim or swarm, for example, and also the bacteria's ability to perform chemotaxis, which is where they move towards certain chemicals. So this indicated that these phages are a very rich source of novel genetic material for Pseudomonas syringae, which could be important for their evolution. Okay, so that's the first part of my talk, which was mainly bioinformatics using our Pseudomonas syringae genomes. The second part of my talk is going to look at how these prophages may contribute towards host adaptation and virulence. And we've done this, and this is a system that I've been working on actually since I did my PhD back in the UK. And this is a pathosystem called the cherry canker pathosystem, which is a really important disease for the cherry industry. And here are some images from that orchard that I showed you at the beginning. And here you can see the Pseudomonas syringae infects the woody tissues of the plants. It also infects the leaf tissues, and in severe cases, it can basically destroy the entire tree. And this is a really important disease because most of the industry here in the US and in other places is moving to these high density systems where you have lots of very young trees packed together in very close proximity. This provides growers with huge yields, but however, it is very conducive to the spread of bacterial canker. And in this orchard that we visited, there was a large proportion of trees showing this dieback, and the grower was very unhappy to hear that they probably were going to lose a significant portion of their orchard to this disease. But bacterial canker It's also interesting from an evolutionary standpoint because it's caused by several unrelated clades of Pseudomonas syringae. And here are 5 of them here. And I'm going to talk a bit later about one called PSM race 1. And these are different strains of Pseudomonas syringae from different phylogenetic groups. So they have convergently evolved to cause this disease, and they're all very distantly related to each other. And if we look at the core genome phylogenetic tree that I showed you earlier in my talk, what we find is that the ability to cause disease on cherry seems to have evolved multiple times. And here are these groups that have been defined, as I said earlier. So these different strains have convergently evolved to cause disease on the same host. And this was actually the topic of my PhD like 10, 11 years ago, because we thought this is a really interesting system. So all of these strains have convergently evolved to cause disease on the same host, and therefore, do they show patterns of effector gain and loss, which is associated with this common host? So do they show effectors that are in common with each other, which are important for causing disease on cherry? And also I explored, do they lose or mutate effectors because these could be recognized by the host immune system? So we thought that this is a really nice system. Not only is it has industry importance, but it also is a really nice system to look at the evolution of host specificity. And what we found was that HOPAR1, which is a type-free effector, which has been really well characterized in Arabidopsis. It's a cysteine protease effector, which acts at the cell surface to suppress immunity. We find that this effector was found in most of our cherry pathogens, and it seemed to be enriched in strains that were pathogenic on cherry. And often what we found is that groups, for example, here, the cherry pathogens possess HOPAR, but strains closely related to them didn't. So it appeared that the cherry pathogens have acquired HOPAR during their evolution. So in order to look further at this, I wanted to see where the HOPAR1 gene was in the genome of these cherry pathogens. So I took the HOPAR1 gene, and then I found its genomic location, and this is what I'm showing you here. And these are 4 different cherry pathogens. And what we find is that the HOPAR1 gene here in red is often associated with phage sequences, which are present here in the green. So this indicated that this effector in these different cherry pathogens seems to have been acquired on a phage during their evolution. And what was even more striking is that if we take these phage genome sequences and we look at where they are on our VIP tree phylogeny, so this is our phage phylogeny, we find that some of these phages are actually very divergent from each other. So the phage in this strain is present here, and the phage in this strain is present here. So this indicates that the same type-free effector has been gained on very different phages. So their relationship with different phages has allowed them to acquire this same effector gene. So what I'm exploring now in my lab, and this is what I'm going to go through with you, and this is very preliminary data, is all unpublished data, is what is the functional significance of these prophages in our cherry canker pathogens. So this is work that's being done by my postdoc, Daniel Maddock, and he joined me just over a year ago. And what we wanted to do is work out how this prophage, does it contribute to virulence in any way in our strain? So we're working with a cherry pathogen, as I said, called PSM race 1, and that has a full phage within its genome, which carries the HOPAR1 effector. And what Dan did is he made a mutant of the bacteria where he removed the entire prophage region. So he removed this entire region and created a prophage deletion mutant. He then did pathogenicity assays on cherry in order to see whether this mutant that he had created showed any difference in virulence. And what he found was that the wild type was able to cause disease, whereas the prophage 1 deletion mutant was actually less able to cause disease. And when he extracted bacteria and ground them up and counted them on agar plates, in order to look at the bacterial fitness and their ability to grow in the plant, he found that the prophage mutant was able to grow to lower levels than the wild type. So this indicated that the prophage mutant has a lower virulence, and therefore the prophage may be contributing something towards the Pseudomonas' virulence on cherry. We also have a mutant where we deleted the effector HOP-AR on its own. So this pink gene up here, we just delete this effector in order to see if the effector which is contributing to this virulence effect. However, when we just delete the effector on its own, it doesn't show any significant difference in its ability to grow on cherry leaves. And therefore, it indicates that something else on this phage or some combination of genes on this phage are allowing the bacteria to grow to higher levels and be more virulent. So we now wanted to look at what genes on this prophage confer this benefit, this virulence benefit to the cherry pathogen. So what Dan did is he looked at the prophage, and this is a schematic of the prophage region, and it has 136 genes. So this is quite good. This is a relatively small number of genes that we can work with. We're no longer working with thousands of genes. And what we found is that after the standard annotation, gene annotation using BACTA, 63% of these genes were labeled as hypothetical, which is not very helpful. So then what Dan did is he took a program called AlphaFold2, and this produces a prediction of the protein structure. And with this, we can look at more remote homology. We can find, by predicting the protein structure, we can work out what other proteins it is similar to, and therefore have some kind of prediction on what its function might be. And what we find is that after the AlphaFold2 homolog prediction, only 7% of the proteins were left unclassified. So this is a really great tool, and I highly recommend. And what we found was that we have a lot of phage genes, so genes involved in producing phage particles. We also have multiple virulence genes, which is, we have HOP-AR, which is present here, but we have other virulence genes, which we're exploring. We also have multiple defense systems, which is really interesting. So these are these immune systems that provide the bacteria with defense against other mobile elements and other phage. And we find at least 4 or 5 different transcriptional regulators, so transcription factors, which could be influencing gene expression in the bacteria. So what we're exploring is if the prophage can alter the expression of other genes in the PSM race genome. So we're looking at these transcription factors because maybe they are involved in changing gene expression in some way, which enhances the virulence of the bacteria on cherry. So this is very preliminary, but we've done a very early transcriptomic study. And the four of the transcription factors are quite interesting to us. So the first two, COM-R and LuxR, are transcription factors which are involved in quorum sensing and biofilm production. So we think that these could be important. These phenotypes are important for virulence. So these transcription factors may be influencing the bacteria's gene expression in some way to enhance its virulence. We also had a transcription factor belonging to the TET-R family in animal pathogens. TET-R transcription factors have been shown to be involved in virulence. And we have an uncharacterized transcription factor, which is homologous to a ribbon helix transcription factor, which we don't know what it does. So what Dan did is he took his wild type strain and his PP1 mutant strain, where he'd removed the prophage, and then he grew them in a minimal media, which replicates the plant environment and induces the production of virulence genes. And then he did an RNA-seq in order to look for differentially expressed genes between wild type and prophage mutant. And the first thing we wanted to do was look at our prophage region within the wild type to see what genes were expressed. And here we have a schematic of our prophage, and then this is the number of reads from the RNA-seq. And what we find is that, firstly, the lytic pathway, so the pathway involved in producing new phage particles, was mostly these genes were switched off. So this indicates that the bacteria are mostly in lysogenic form, so they're dormant and they're not actively producing new phage. But what was really interesting was that the transcription factors, which I've highlighted across the graph, were very highly expressed. And so they were some of the most highly expressed genes on this prophage. So we were thinking that they're highly expressed, so they could be influencing expression of other genes in the genome. So what Dan did is he then, after removing the genes in the prophage region from his analysis, he looked at other genes in the genome to see which ones were differentially expressed between the prophage mutant and the wild type. And he found 131 genes were differentially expressed, with 100 upregulated and 31 downregulated. And what this word cloud shows, this is the predicted functions, is that we find that pathogenicity was one of the highest categories of functions. So this is really encouraging. It indicates that the prophage may be regulating virulence gene expression in a way that is important for pathogenicity. And this is just another way of presenting the data. So these are the 131 genes. And what he found was, and this is their log 2-fold change between the prophage mutant and the wild type. He found that, for example, genes that were downregulated in the PP1 mutant included genes involved in heavy metal resistance. And the fact that they were downregulated in the mutant indicates they're upregulated in the wild type. So the phage may be providing resistance to the bacteria for heavy metals. And then in terms of genes that were upregulated in the mutant, he found genes involved in chemotaxis, type 3 secretion, type 6 secretion, and amino acid biosynthesis. And this was quite interesting to us because these genes were upregulated in the PP1 mutant, which means that they would be downregulated in our wild type. And this was a bit confusing because we were like, oh, this is kind of the opposite of what we expect, but that is science, I suppose. Because what we found is that the type 3 secretion system seems to be upregulated in our mutants. So maybe that indicates that the prophage may downregulate its expression, which is kind of not really what we expected, but it indicates that the prophage may be regulating the production of this really key virulence factor. And this is where we're kind of up to, and I've nearly finished, just a couple more slides. So we know that in some way, through our RNA-seq, that the prophage, PP1 prophage, is influencing the expression of the type 3 secretion system. And it seems to, in our RNA-seq, have suppressed the production of the type 3 secretion system. However, we know that prophages are highly dynamic mobile genetic elements, and maybe they shift their transcription a lot during their activation. So we're now interested, our RNA-seq was just a very preliminary one-time point bulk RNA-seq. So we're looking at the whole population at a single time point, and we really need to look at this further to understand what's happening. And if we look back at the phage, we know that it will have a lytic pathway and a lysogenic pathway, and maybe when it's induced and when it starts to express this lytic pathway, this could alter the gene expression of other genes on the phage, such as these transcription factors, in a way that will affect this repression or this relationship. So if we think that this phage may be allowing the bacteria to tightly control the production of its type 3. So we're now exploring this further. We want to examine the gene expression of the genes from the phage and how they influence the global gene expression over multiple time points and try and understand what happens when the phage is induced. And we're creating strains where we're tagging them with fluorescent proteins to monitor how the phage gets activated in different conditions. And I just wanted to very briefly come back to this phylogeny. So this is the VIP tree phylogeny of Pseudomonas phages, and our PP1 phage that we're studying is present in this genetic group 4. And if we on our core genome, this is the bacterial phylogeny, if we put we put this data onto the phylogeny, we find that this genetic group 4 is actually present across the whole of the Pseudomonas syringae complex. So this means that the results that we're seeing and that how this phage may be regulating different genes in the bacteria and contributing to virulence could actually be conserved across other pathosystems, which is really exciting. So this is kind of a schematic of what we're working on. So we know that these phages are involved in horizontal gene transfer, and they may be moving type 3 effectors around bacterial communities. And these prophages carry multiple accessory genes such as transcription factors, which we think may influence virulence gene expression and alter pathogenicity on the plant. And we now want to understand how these various factors, such as abiotic factors as well as plant factors, will influence the activation of the phage and how this leads into how this affects its role in virulence. So it's really, we're just starting these projects, and it's really exciting, and we're hoping we can update you later. Okay, so I'm just going to acknowledge the people in my lab. So Daniel Maddock was a postdoc that did most of this work. Also, George Sundin is my collaborator, Professor George Sundin here at Michigan State, and he lets me go into his orchards and collect lots of Pseudomonas. This is my lab group, and here are the funders that funded this group. And we really enjoy working in the field. And here, obviously we like our lab work, but it's also cool to go out in the field. And here's a picture of a raccoon that we took last year. And as a British person, having moved to the US, we don't have raccoons there. So this is quite exciting to see a little raccoon hanging out in the cherry tree and watching us while we do our sampling. And thank you so much again for the invitation, and thank you all for listening.